Let's talk about, again, I'm talking mostly about advanced melanoma here, because that's what I study as a medical oncologist. So here are the FDA-approved drugs for melanoma. Of course, the FDA is the Federal Drug Administration that sort of decides what a doctor can give a prescription for. And I think it's worth noting, so in blue are the drugs that were FDA-approved without documenting a benefit in how long people actually live. They documented a benefit in how long the drug took, the tumor took to come back when the drug was given. Then, as you see, nothing between the 70s and the mid-90s, and then a long sort of gap, and now sort of an explosion. And so we've talked a lot, or a little bit earlier, about ipilimumab, pembrolizumab. These are immune drugs. These are targeted agents that block certain proteins in cells. Now, these are the FDA-approved drugs specifically for uveal melanoma. Right? So, why is that? Well, we've had a lot of trials that were run by a lot of really smart people using the smartest things that we had at the time. Sort of look, RR means how many people had enough shrinkage to sort of call someone definitely responding. There's a certain cutoff. So not everyone who has a little shrinkage counts. But very few people using sort of these agents had a lot of shrinkage. And so that sort of tells you that you're not going to be able to get your drug approved, and it's not really showing a benefit for patients overall. And so, of course, this underscores the point that uveal melanoma is not skin melanoma. So when I say uveal, what do I mean? Well, earlier the ophthalmologists have said, you know, the uveal tract, they call it choroidal melanoma. It's composed of three things. I stink at the back of the eye anatomy, and so I like to use one word that encompasses the choroid, the ciliary body, and the iris. And so I just call it uveal melanoma. Someone earlier said, you know, within ocular melanoma, don't forget about conjunctival, and we haven't. It's just that genetically, conjunctival melanomas actually are very different genetically. What we do know is that almost all uveal melanomas, not skin melanomas, and not conjunctival melanomas, not melanomas that start in the mouth or around the vagina or the anus, all of these tend to have mutations in either GNAQ or GNA11. I'm going to talk a lot about that. We know that they're different because they act differently. As we've talked a lot about, 90% eventually will go to the liver. Our numbers suggest maybe about 85% is the first place it goes. So that's why we've all been talking about liver-directed screening. This here, you know, I firmly believe in this statement. And what does this mean? The standard of care that we always talk about is sort of what can we stand behind and say there's unequivocal proof, taking half of people, randomizing them to either a sugar pill or not, and showing benefit. And as I said, in skin melanomas, we have several treatments now in the last few years that have shown that benefit. We haven't yet done that in uveal melanoma. So I'm not saying a clinical trial is best, but what I'm saying is that's the only thing we can stand behind and say we have proof for. We have proof for nothing, and so we have to continue trying to prove. Does that make sense? So I wanted to talk a little bit about the genetics of GNAQ and GNA11, and I apologize, I was supposed to add this in. So GNAQ, oh yeah, I did actually. So the purple, the alpha here is what I'm talking about, and it's these two genes, we all have them in our normal cells. This is how we live and grow. These are all normal things. And so normally there's no signal for this protein. This is the outside of the cell, this is the inside of the cell. Luckily, you've heard all about sort of these protein signaling pathways earlier, so my job is a little bit easier to say you get a signal here, and this, this GNAQ or GNA11 that usually holds on to this molecule here, this is just like if you're a runner, you know, those goo things, those like little sucrose things that you can eat along the way. It's sort of concentrated energy. And so it's holding on to concentrated energy here, and normally with a growth signal there, you get a sort of concentrated energy packet, and then it separates, and the alpha protein starts to send signals to other things to grow. And since it has this energy packet, it can pass it along, and this is how our body sort of gives growth signals. Well, normally, there's a couple of spots on the protein itself. So these red little fingers are my way of not scientifically trying to show a nice little pocket for the energy packet. So these are two normal amino acid residues, the part that makes up a protein, one specific spot or the other specific spot. And normally, over time, after it signals for growth, it can turn itself back off. There's some other regulators of signaling that attach, and the energy packet sort of runs out, and so GDP goes off, it meets beta and gamma, and kind of goes back. So it's kind of like this. That's normal. When you have a mutation, which we know almost all UV melanomas do, you're sort of stuck in the on position. So the normal R183 or the normal Q209 position gets a misspelling. And the misspelling comes from a mutation in the DNA that turns the protein into a different sort of configuration. So this is normal, but let's see if this works. Yeah, so normally this happens, but when you have a misspelling, you can't turn the signal off. And so it doesn't go back to the off state, and what you have is this signal with the energy packet stuck in there, signaling downstream. And the big question is, of course, what are these signals downstream? Because if they start, all these UV melanomas start with the mutation. If we understand what it's talking to and what the on signal is, where the on signal travels, we can try to maybe cut that signal at that point. So simplistically, it's kind of like a string of Christmas lights. And if you can figure out where those lights are going, maybe you can take one of those bulbs off, and maybe it'll turn the whole chain off. And so this is just a review of the cell signaling. This isn't like I did all of this, so I want to make sure you guys don't think I'm some kind of genius here. But walked into a situation where our group and a lot of others knew that this protein chain called the MAP kinase pathway was turned on in these cells, and nobody really knew how. But there was a group out actually at UCSF and some others that showed that these mutations were there, and so they seemed to talk through protein kinase C, RAF, MEK, ERK, and then into the nucleus for growth. So the idea was, if we had a drug that blocked one of these proteins, and for a number of reasons they chose MEK, like selumetinib or a cousin drug called trometinib, then maybe we could cut this whole chain off, and people will have dramatic reductions in the size of their tumor and things like that. That's the hope. And we had some reason to hope for that, because in skin melanomas, there's a BRAF mutation. Instead of a mutation here, there's a mutation here in about 60% of cases. And so they had some nice reductions with a blockage of this protein. And so that's the idea for the trial that my mentor, Dr. Carvajal, just recently published. This started, to give you an idea, the protocol number for this, the first two digits are the year, and it's 10053 at my institution. So this started in 2010, and we just released these results about 4 months ago. So it tells you how long these things take to sort of understand. What we did was we took people with uveal melanoma that had spread, and we said you either get a chemo called DTIC or dacarbazine, or you could choose temozolomide, or you get the MEK blocking drug selumetinib. And a computer decided, it wasn't the doctor's decision. That's called randomization. And then if things grew on regular chemo, you had the option of getting the selumetinib. This was an important trial for a few different reasons. One is no drug had shown benefit, no pill therapy had shown benefit in a randomized way where you couldn't choose who to give the pill to. You couldn't just cherry pick and say, oh, that guy's going to do pretty well, I think. I'm going to show that this pill does really well. A computer picked what you got. This was the first trial to really, as I'll show some benefit in even the growth of the tumor. So that's called a progression-free survival, how long you can go without your tumor growing a certain amount of time. So this is the way that we always show these data is how long does it take for the tumor to grow. And so if your tumor doesn't grow, you're still counted in this progression progression-free. And so everyone starts at 100%, of course, and then you get scans every 4 to 8 weeks. And we saw that selumetinib, the MEK inhibitor, slowed the amount of time that it took, slowed the growth of the tumor, so that instead of on average 8 weeks for chemo, it took on average 16 weeks. Now, if we stretch this out, you see that this hasn't gone to 0. And so what I'm not showing you is the rare patient who's actually out 1 or 2 or even about 2.5 years without any change in the tumor growth. And that is really exciting because number one, on average, we're showing a benefit, which is one of the first things you need to do to make progress in a field is randomize and show a nice benefit to this drug over what you currently have, which is regular chemo. The second is I tell patients you don't want to be average if you have metastatic disease. You want to hope that you're in the good side of the average, right? Stats are just stats. You're not 50% progressed or 40% progressed. You've either progressed or you haven't, right? So if you are in this group that benefits, that's all the difference in the world. And if you're a person who doesn't benefit, you know, you're going to throw eggs at me right now because it didn't matter to you, right? So on average, we're excited about this because we learned that people benefit. And this is one other way to show how people benefit. Each dot here is a person, and on this axis here is how their tumor did while they were on the study. So if your tumor grew, you're above the equator. If your tumor shrunk, you're down here. And so it doesn't take a scientist to understand that chemo didn't seem to help as much as the MEK did. So you see about half of patients had some degree of shrinkage for some period of time. Of course, there's no free lunch in this process, and so we'll be the first to admit that there are always side effects of being on the drug. And the way it works on a trial is people will ask you every time you come in, do you have any fatigue, any weakness, any this or that, or nausea, and all these things. And then we check blood, of course. And so there are two things here. I purposely didn't want to make this readable. I just wanted you to know that there were a lot of side effects in general, but that most of these were what we call grade 1 or grade 2, and the grade goes from 1 to 5. And typically, so grade 5 means you died from a side effect. Grade 4 means generally you were hospitalized. Grade 3 means pretty close to being hospitalized or at least significant enough to change something. And then 1 and 2 is sort of, we notice it, patients notice it, but it's something manageable. So the vast majority of this stuff wasn't really grade 3. So the things that were grade 3 were blood tests generally, and we watch these things, liver inflammation, this muscle inflammation, and these generally got better when you stop the drug or hold it or lower the dose. So not a free lunch drug, but what did we learn from this trial? We learned that this is the first time we've been able to show, without cherry picking who we give the drug to, that the one drug is better than another. The other thing we showed that I didn't even put on here is that it really kind of, I think, and again, I'm not even on the paper, to be honest. I have like no truck in this. I think it kind of galvanized the medical oncologists to sort of say we can do a study with multiple sites. We can do it in just uveal melanoma. We don't have to piggyback off of skin melanoma and try to say, well, 10% of this study had uveal, so maybe. No, we can actually design and execute a trial specifically for people like yourselves if you ever need it. I think the reason I'm standing up here is that, of course, almost everyone, if not everyone, if you live long enough, will have tumors outsmart the medicine. So the question is, what is doing it? If we're blocking this effectively, then what are the other sort of escape pathways? And so how do we do that? Well, we can select for cells in a Petri dish that grow despite the MEK inhibitor. So they grow on plastic, you give them the drug, most of the cells die, but over time you can find some cells that have evolved to resist it in the lab. The other is, can we study from actual patient tumor biopsies from the selumetinib study to see what changes they actually had? So this is out of my original mentor's lab, Gary Schwartz's lab. They looked at cells in the Petri dish first, and what this is these are two things. This is called a Western blot. All you have to know is that if you have protein present, it's dark. And so here there are two cell lines, OMM and MEL270. They made them resistant over time. That's what the RS is for. So these are cousins, but one is resistant to a MEK inhibitor. Same thing here. And they saw that without anything, OMM1 doesn't have any of this protein called phospho-AKT. When you make it resistant to MEK inhibitor, it does. Same thing with MEL270. It has a little bit at the beginning, but it certainly has more when you make it resistant to selumetinib, which again blocks MEK. So the thought was, is AKT one of the reasons for resistance to MEK? And so one of the ways to test that is you add a blocker of AKT and then say, does that make a difference in growth? And here you see when you add, the plus sign means we put the AKT inhibitor or blocker in. And you see, okay, nothing there, so that's not unexpected. When you have AKT there and you block it, look, it goes away, look, it goes away, look, it goes away. Well, let's see how they live and grow on a Petri dish. It looks like the OMM1 and MEL270 by themselves, these are the two up here. Trust me, because you can't really tell by the grays, it's kind of hard. But when you give more and more of this AKT blocker, you only see a little bit of decrease in the number of living cells. You see some, but this is a fair amount of drug, 5 micromolar. Now, if you make them resistant, where they've outsmarted MEK, and you add AKT, you see a much more dramatic decrease at much lower doses of the AKT inhibitor. So the idea is, there's this whole other side of the pathway that I haven't even drawn what the receptor might be that signals to it. It may be a number of things. But something's signaling when we try to block MEK. All right. When we try to block MEK, something may be signaling here. And so later in the same project, we checked for levels of, as Dr. Sabula said, the RNA levels, which is sort of the message that takes the DNA to the protein. And we saw that for people on the selumetinib trial who had a response, they had a much lower level of this RNA called DDX43. I'm not going to test you, but I'll show you where it might fit in, okay? So the not responders had a much higher level of the protein. And it's pretty much the same. This is pretty much what this is. This is just showing you 3 different people who responded or 3 different people who didn't respond. And this is different than a Petri dish. This is actually asking our patients while they're taking the pill to say, well, 2 weeks into the study, we're going to ask you to get a biopsy. And I want to just thank you guys, or anybody who's been on a trial who's gone through this, because this is what this allows us to do. It allows us to look at the protein level in the body. You take it out, you freeze it, and you study it, what the proteins are doing. And here you can see this DDX43 in general, now it's not perfect, but in general is higher in those who don't respond. And ERK is the protein after MEK. So if we blocked MEK, which we do here, it goes away, goes away, goes away. Oh, doesn't go away, doesn't go away, doesn't go away. It sort of suggests that there's a number of different things that are talking to ERK and talking to the other pathways that are outsmarting the selumetinib. And it may be DDX43. I'll skip ahead a little bit. It may be these 2 proteins here that are in what's called the RAS family. And so here the idea is that if we block here, this is what the model suggests, and again, this is kind of simplistic, but we block it here, But meanwhile, there's sort of this escape pathway that goes through this protein, talks to these proteins, and it increases these AKT levels. And meanwhile, I mentioned things being like little bulbs on a string of Christmas lights. But when's the last time you ever took out one bulb on a Christmas light and had them all go out? Unless you bought really cheap. Those stopped maybe 15 years ago. You know, now they're all in parallel. You take one bulb out and they're all sort of still firing. So this is a little bit what we're seeing when we give selumetinib. Some people have the entire string of Christmas lights go out, but most people don't. And we wish we sort of knew why that was, but that's what we're sort of working on. And so the idea is then if we give a MEK blocker, could another way to benefit be to add a blocker of AKT? And this is one of those like, you know, alphanumeric codes for a drug before it has an English name. So the research questions that we've embarked on is, can we combine blocking MEK with AKT to improve the control and shrinkage of the melanoma? And then just as importantly, can we understand, take it back to the lab and understand how these uveal melanoma cells outsmart either MEK blocking by itself or even both of them? So how are we doing that? These are what we call the aims of the grant that, again, I was fortunate enough to have funded. One is to help along with the phase 2 multi-center trial. This trial is, as I'll mention, open you know, at our center, MD Anderson in Houston, and we just welcomed in Nashville, it's escaping me, oh geez, Vanderbilt and Emory just on Thursday, or Wednesday. And then we want to get biopsies to evaluate markers of sensitivity. You can do all you want in a petri dish, but you really got to look at what's actually happening in a human being to try to make more progress. And so this is the trial that's underway. Kind of like the other one, it's randomized 1-to-1. On one side, you're getting just a MEK inhibitor. It's not the same one. It's the cousin drug trametinib. And then on the other arm, it's the MEK inhibitor trametinib with, we've shortened the name, but it's the same one, the AKT inhibitor. And then if you grow on just the one, you can add the second one. Again, we're looking at how long it takes for tumors to grow. We are going to follow people to see, you know, how long everyone survives, whether it's safe and tolerable. And we want to see, in general, how we can do when we block not just one signal, but 2 signals. There's going to be up to 40 in each arm, and so that brings us to 80 total. So where are we? I was very firmly told, you know, it's too early to give results, and I know that's really what people want to know. We honestly just don't know enough. What I can tell you is that 23 out of 40 patients have enrolled in just under a year, which is great. 19, I'm very excited about this, 19 out of 23 people were able to get a biopsy at the beginning and after 2 weeks of being on the drugs. And those are very important because we can ask what genes are on, what proteins are up at the beginning, and what happens after 2 weeks of blocking either just the MEK protein or blocking MEK and AKT. And the fact that, you know, 19 out of 23 so far have gotten biopsies is really exciting, because it's not always easy in real life, and it's not always safe, and it's not always, you know, fitting into somebody's schedule to get it done. So honestly, that was it. That's the data update you get from me. So I wanted to just call attention to the fact that obviously the entire field does not boil down to MEK, and there's a ton going on that that I want to just highlight. So what are the other signals? We've kind of talked about some. This is the figure. This is the figure that, you know, as we published it. So there's this side. I didn't even want to get into this because this just came out about 6 months ago, but it's exciting, and it came out of a couple labs, one at the NIH, one at UC San Diego, where this sort of protein called YAP seems to be active in uveal melanoma cells. And so that gives us a potential second or another target, another potential weakness that there's nothing up and running at this point, but this is certainly an area that we're excited about. Not just us at Sloan Kettering, I mean we as a field. Well, Alex, if everybody has mutations in GNAQ or GNA11, then why is only that, some spread? Why am I the unlucky one and other people are lucky? And this is just a smattering of the other genes that are involved. I'm not going to spend a lot of time on this. There's a lot of other stuff going on. Earlier we talked about things that were so-called epigenetic changes, so not the spelling of the DNA, but maybe whether it's to use the analogy, if you read an alphabet and it's A, B, C, D, you can, without changing the letters, you can make it easier or harder to read that word by bolding it or making it the same shade as the background, so to speak. And that's what I think of as epigenetic changes. So there's a lot going on there. Can we prevent spread? I'm not going to spend time on this either because we talked earlier. The Cleveland, I'm not Cleveland Clinic, but here in Ohio, Case has done this. We're waiting on results. We have it open, and we're happy to have the crizotinib study open at Ohio State fairly soon. And can we activate the immune system? That's another big area of study. This, I think, is a useful way. I know we've talked a lot about this stuff, but again, to look at a T cell, which is your white blood cell that kills tumors, there are things that help it turn on and try to kill things. There are things that inhibit it because we can't just have it attacking everything in our bodies uncontrollably. And so the idea in a cancer scenario is you want to either turn up the activating side or you want to block the inhibitor side. And so we know some reports from ipilimumab, which blocks this one here, CTLA-4. We know that there are some patients, again, the response rate is fairly low, but we're more interested in how long the disease is controlled by your own immune system. There are some patients that are controlled at 3 months, which doesn't sound like a lot, but if they are controlled, they can sometimes be controlled for many months, or on the order of 1 to 2 years. Someone earlier said some are brave enough to use the cure word. I would not be one of them. I think, again, I'm a stodgy person who wants to wait and see how people do, but it's certainly very, very exciting. People ask all the time about PD-1, which is this here, again, blocking the inhibitor side. And again, we have no published data. So in the end, coming into this as a young guy, I was joking around earlier, I haven't been around long enough to be depressed about treating melanoma. And I think that's what happened, for everybody who's paved the way for people like me, is they did a lot of hard work and they had the hard conversations with patients about a lack of options. And that's not where we are now. We have an explosion. And so we have a lot of studies going on. Blocking MEK helps. Blocking MEK helps. We're really trying to make it better. And there's reasons for hope soon, but of course, it isn't soon enough. It's never soon enough. So with that, I want to thank my melanoma service at Sloan Kettering. My former mentor was Gary Schwartz. He left. Rich Carvajal took over. Now he left. So I just have to work with 3 or 4 more people, and then I'll be the head of melanoma. Well, they all got promotions, so they left for good reasons. These are our participating sites for the study I talked about. I want to thank again the Ocular Melanoma Foundation. I also have for another project some funding that I have to acknowledge from the Conquer Cancer Foundation. So thanks very much.